Of a body, how to research to write the review, and in the case that you want to analyze the data quantitatively, how to do a meta-analysis. So that's what we're going to be doing, and this is sort of, you know, a very informal primer to this. So if there's things that you're uncertain about as we go, just please shout out questions, interrupt me. I'm happy to clarify, or if you want to make some comments on things that you have experience with, please. This is not meant to be a formal seminar. All right, so let's see if I can not get myself to the next slide. Here we go. I think it's useful to reflect a little bit about the history of meta-analysis. It's been used in ecology and evolution for really only a couple of decades, but it has a much longer history in medicine and sociology. Those are the fields that really originated this practice, and they've been doing it for ages. And I found this paper that's from 1989 that is like, where are we? Where are we going? That just to show you how old it is in medicine. But it's much younger in ecology and evolution. And I think what we're doing is sort of benefiting from their long history of use to make best practices to suit us. But in some ways, there are some differences, of course. So I just wanted to highlight this. And it arose originally because of the need to synthesize across many independent trials. So doctors are trying out new medicines or treatments, and they want to know, is that more effective than a placebo or some other treatment? And so they want to synthesize across all these independent trials. And the first quantitative approach to do that is just vote counting, right? It's just make a stack of the studies that found that the treatment worked better and a stack of the studies that found that the treatment didn't work so well. That's just vote counting, counting up numbers that yes or no. That is very coarse, obviously. And when we do that, we may get a false sense of the support for a hypothesis or a treatment or a medicine, because the studies can vary, of course, in their rigor, in their sample size, in the magnitude effect that they detected, in the arbitrary threshold that they used to consider the treatment to be statistically significant, et cetera. And so to get a better sense of the overall evidence or support for a hypothesis, people wanted to be able to incorporate those aspects of, you know, study quality or rigor or impact and incorporate that into the synthesis. Of course, we can do that qualitatively, just through our own assessment. This one matters more than this because of all these different features that they used. The problem is, of course, that we're subjective beings, and we're likely to impose bias based on our own experience, et cetera, lots of things. And so it would be better if we could do this kind of synthesis quantitatively. And that is where meta-analysis came in. So the point of meta-analysis is to gather the actual data from those studies, incorporate the magnitude of effect, which in the case of a placebo versus treatment is just a mean difference, but then also incorporate the variance within those groups. That reflects sample size and accuracy. Those are the key ingredients that we can then analyze collectively across a body of evidence using meta-analysis. So that's how it arose. Same thing in sociology, different kinds of groups responding to different kinds of environments or things like that. And now, of course, in ecology and evolution, we're trying to use the same sort of approaches, but maybe our ways of doing science are not so systematic or consistent across scientists. We're not doing all sort of similar kinds of trials of how medicines work. So I think it's gotten a lot messier, but that's also more interesting. Well, I'm an ecologist, so to me it's more interesting. And that's where in the last 20 years, there's been a huge amount of literature trying to hone and adapt these approaches to the complexity of ecology and evolution, which I would say are very creative sciences. That's one of the things I like about them. Different scientists approach the same questions in different ways. To me, that's a real asset, but it does complicate synthesis. So that's one of our challenges. All right. Guess I just have to do like this. Okay, Also, I think that this is a pretty popular thing to do for graduate students. A lot of times you do an introductory chapter to sort of review, get to know the body of work that you mean to be working in, and you might be tempted to then publish that as a review. I think that's a really great strategy. Publishing a review as a grad student is a really good way to get your name out there. These tend to be high impact papers. They get more citations on average than research papers. So that's a really good strategy. I think there are real advantages and disadvantages to approaching a review on a topic that you are not yet an expert on. The advantage is you have less baggage. So you tend to be less biased, I would say, than somebody who knows the literature well. The disadvantage is that you don't know the literature very well, right? So you may not catch all of the literature that you need to synthesize, or you may not know what are the likely factors that I need to keep an eye on across all these studies that may explain why some studies find this and other studies find that. Experience helps us with that. So, of course, science is very collaborative. There's an easy solution to this, which is just pair up with somebody that has the expertise. Done. Okay. The goal of what we're doing today is I'm going to walk you through how I would do a meta-analysis. And a lot of that really in making sure that I have a good body of evidence to synthesize. Anybody can do a meta-analysis on any pool of data. It's not hard. In fact, it's super simple. And it's very tempting because a meta-analysis is a buzzword. So we all want to do this. And it carries this sort of hint of objectivity because it's quantitative. So we're inclined to want to believe them more than maybe a narrative review. But if you haven't been really rigorous in your compilation of the data, then your meta-analysis is not informative and it is subject to bias. So we have to be really careful that we are confident in our body of evidence. And so a lot of meta-analysis is really that. It's being transparent about our methods of compiling the evidence. That's what systematic review is. So a lot of what I'm going to talk about is that, which feels like less exciting because it's not the running the models, but it's the reality of doing a good meta-analysis. All right, I come at this as a practitioner. So I'm a functional ecologist. I started doing meta-analysis when I was a postdoc about 20 years ago in London. I got a fellowship to go work with somebody who's a leader in meta-analysis and systematic review in ecology. So I just got plopped right into it. At that time, there wasn't a ton of ecology and evolution literature. So I read a lot of medicine and sociology meta-analysis books, which was actually really helpful. And I've done it ever since then. I love doing synthesis. I've written 10 different, what I call integrative reviews. Those are not quantitative, but just a classic sort of narrative review. And I published 5 meta-analyses and 1 perspective. So this is a major part of the research we do in my lab. So I do read a lot in this field, but I'm not an innovator. I'm not somebody who's developing best practices or analytical tools. I'm a user. So I'm coming at it from that perspective. There's always more as a user that we can learn. So I think that's really important to acknowledge up front. I would recommend if this is something you're interested in to get this book. This is the book in ecology and evolution. There's Yulia Krycheva. That's who I did my postdoc with. And it's a really good edited book from a lot of leaders in this field. And I think what this helps elucidate is that research synthesis is a formal scientific method. I mean, a lot of times I think we view writing a review as just something you sit down and do. Like you just read a bit and then you write something, but it's actually got expectations of formal procedures like any other discipline in science. And so I think being aware of that and then seeking out those resources so you learn those best practices early in your process of writing a review or doing a meta-analysis is really helpful. Europe is kind of where this is being led. There are whole institutes and journals and departments on research synthesis. So there are a lot of innovators who are sort of working in how we can do this better. And the reason this is so valuable now at this point of time is that the literature is vast. There's so many scientists doing science all over the world. that there are huge amounts of evidence now. So synthesizing that evidence is more and more challenging. And of course, there are more and more smart people thinking about how we can do that better. So these are the people who we need to look to learn these best practices. Okay. Reviews are called lots of different things, right? So you've probably heard review, narrative review, integrative review, systematic review, meta-analysis, perspective, opinion, synthesis. Different journals call these different things. Different practitioners call them different things. I find that there's a lack of consistency in how people use these words. What I would call synthesis is not the same thing that some other journal calls a synthesis. So this is messy, and I wouldn't put too much weight on these terms. They all have the same goal. It's just to synthesize a body of evidence, right? To me, the two key criteria that can define a type of synthesis is one, how you compile your evidence. So the literature, the papers, the data sources, are you using formal systematic approaches to that or not? And if not, that's the old school way of writing a review where you're just like, I've read a lot of papers on a topic, I just gathered them all together. That's not systematic. There's a formal systematic procedure for doing this, and that is the first distinction. The second distinction is what you do with the body of evidence. Are you just going to assess it qualitatively, vote counting, or a sort of interpretation of the studies? Or are you going to actually extract the data for a formal meta-analysis? Those are the two things that we would want to know about, and it could apply to any of these words, except that meta-analysis is the formal procedure for quantitative assessments. Otherwise, well, I'm going to come back to perspective and opinion in a sec. The point of systematic review methods for compiling the body of evidence is that it's meant to reduce bias. This is obviously part of science. You can find narrative reviews on the same subject by two different scientists that draw different conclusions on the same topic, right? I'm sure we've all found this. And the question is, why? Why are they drawing different conclusions from the same body of work? That's because of their bias. They're weighting different factors differently. They like something about this approach and they find that evidence to be more compelling. They take their own work to be more compelling. I don't know, there's lots of reasons why bias could come into play. The problem is that that's hard for us to interpret because we don't necessarily know what bias is factoring in. So the point of doing systematic review is to try to eliminate that bias by being as transparent about our methods of how we compile the evidence and hopefully being less subjective in how we do that. So some of the reasons why this can come up is that people exclude relevant literature, maybe just unintentionally because they didn't find some papers. We can't assess that unless we know how they searched for papers. So knowing how they found papers is really important. They may have excluded papers like explicitly, intentionally. And again, if we don't know that when we're reading the review, we may not know why those papers weren't included. So transparency in which studies were included and which studies were excluded is really helpful. That's part of systematic review. A major source of bias in synthesis is associated with language, right? So many people who are publishing in science are using English, but there's still lots of journals in science in the world that aren't published in English, of course. And if we're excluding all of that, then we're excluding a large portion of the literature, and it's not random biogeographically. So we're probably missing some of what's relevant to our study because it's not in a language that we speak. And then, of course, there's publication bias. So null results, negative results would factor into publication bias. Sometimes scientists work for the government or in agencies and they don't publish in peer-reviewed publications, but they publish as reports. If we don't know about that, we may be missing an important source of data. So that's something that should be considered when compiling your evidence. Systematic review should help us address all of these issues. Okay. Now I've taken those different names for reviews, and I've tried to put them in what I think is these two factors. How did they find the literature, systematic or not? And how did they assess the evidence quantitatively or qualitatively? So this is where I wanted to point out that perspectives and opinion review articles are intentionally non-systematic. The bias is implicit or explicit in those types of reviews, right? They're arguing a point. They mean for it to be biased. That's in the name. And that's totally fine. That's a legitimate type of science. We just know that that's not systematic. We know that there's probably some literature they excluded. But that's okay because that's the point of those. Everything else should be done with systematic searches. Historically, it came to meta-analysis first. And that's because meta-analysis has this air of objectivity. So compiling all the relevant literature was really important. But narrative reviews were sort of slow on the uptake. And it isn't until maybe the last 10 or 15 years that most journals expect systematic review methods for all review articles. Most review articles now cannot be published without the methods reported in them. How the literature was searched, what was the inclusion criteria, what was the assessment of bias, etc. So I would say everything else should be following systematic review methods. Then, whether or not you analyze those data qualitatively or quantitatively differentiates all of those types of reviews, which are qualitative. So it's just my own interpretation of them. And that can be really important and useful. Maybe you don't need to do a meta-analysis for everything. Maybe the data are variable enough. We're trying to synthesize across disciplines where it's not conducive to meta-analysis. That's all legitimate. But we acknowledge that that's a qualitative review. The meta-analysis is quantitative and is usually argued to be the least biased because we compiled the data using systematic review methods and then we're analyzing it quantitatively. Okay. Oops. Uh-oh. So here's what you do when you want to write a meta-analysis or review. First, you ask what your question is. Then you decide what's the body of evidence that you're synthesizing, and you pull all that evidence together, and then you actually run the meta-analysis. Running the meta-analysis takes like 5 minutes. It's so fast. The rest of the process takes forever, but the actual modeling of those results is really fast. So that's just sort of the cherry on top at the end. I really caution you to ask early if a question is good for meta-analysis. Not every question needs meta-analysis. I think we assume we should do it because it feels better somehow because we've got statistics in there and it's sort of got this objectivity, but not all reviews need to have quantitative assessment. And in fact, it's not that useful if a body of evidence has already reached consensus. So to me, this is what I'm really looking for. If there's a question where the person can say, seems like there's no consensus. Sometimes it's this and sometimes it's that. That's the question for meta-analysis because there's variability in the evidence. And the whole point of meta-analysis is to test potential sources of heterogeneity using statistics so that we can conclude factors associated with the yes or no response, right? If you have a body of literature where it's like, yeah, I mean, it seems like everyone basically agrees it's like this, then the meta-analysis is just going to confirm that, and it's a lot of work. It's not going to be that helpful. It doesn't change anything about how we understand that topic. So I would argue that meta-analysis is most useful in a scenario where there's an apparent lack of consensus and enough case studies that we can investigate potential drivers of that lack of consensus, the heterogeneity tests. Okay. To do the first two parts of this, the setting up your question and finding your evidence, these are your steps. You identify your audience and your field standards because actually different journals have really different expectations of what you need to report. You want to know that early So look at your target journal and see if they have criteria for publishing a systematic review or meta-analysis. Many now do. Define your question and scope. Scope is like your inclusion criteria, basically. What are you trying to focus on and what are you excluding? Plan your search methods like you would any research project. This is methods just like anything. You need to set this up ahead of time because you need to report all of this in your paper. So you can't just muddle your way through this, or if you do, it's done badly. That's the last point. Track everything. Take notes like you would if you were doing a field study. You need a notebook. You need to make detailed notes of every decision you make in gathering your evidence and how you analyze it, because those are your methods that you need to publish with your paper. Trying to remember what you did retrospectively is, I mean, we all know you wouldn't do that for a research project. You shouldn't do it here either. Okay. This has been discussed a lot in the literature. It's kind of related to parachute science. We shouldn't feel compelled to write reviews or do meta-analyses on topics for which we are not a member of that community, right? We need to be really careful about stepping on toes, inserting ourselves into a space where we have no right to be there. And this is discussed a lot with respect to indigenous data sovereignty, but it's also just discussed in general in conservation, because conservation is very place-based. And so when people try to synthesize evidence for a place they're not familiar with, it doesn't come across very well. So the recommendations is always think of who your audience is, who are you trying to reach with your synthesis, and engage them in the process. They should be part of your author team. You shouldn't be approaching them after you've done it to be like, is this useful to you, right? We want co-produced synthesis. And that is going to strengthen, of course, what you're doing, because you're then have people who are really familiar with that body of literature. And it's also going to just be more equitable. And I think that that is, depending on the kind of work you do, really relevant or maybe not relevant. I don't know, but something to think about early. Okay. I would say setting your inclusion criteria, which is your scope, is one of the hardest things. And if you pay attention when you read meta-analyses, this is how you know how far you can extrapolate what they're finding. And this is also a way people cheat. I've seen a lot of meta-analyses where they have really strongly worded conclusions in the abstract, and it sounds like, wow, that's amazing. And then I look at and they only included maybe 30 studies in their meta-analysis because their inclusion criteria were really narrow. And I'm like, okay, well then you shouldn't extrapolate outside of that. You've made a very narrow selection of studies that you think are conducive to your question, but then that's your scope, right? We do this, or people do this, because it's a way to cut back on the labor of data extraction. If we can make narrow inclusion criteria, then we only have to deal with 30 papers, which is fine. You are transparent about that. We just have to then be careful about how far we extrapolate. So setting your scope up front is really important. How big or how narrow do you want this to be? What's the biogeographical scope? What are the study approaches? Are you just doing field studies, just doing lab studies, just doing studies that are well controlled? What are your factors or treatments of interest? So these are the things that could be driving variability across studies. Like in my case, where we study a lot of drought. So is it experimental drought? Is it natural drought? Is it pulse drought? Is it pressed drought? We have to specify this early. What kinds of data are we interested in? Survival data, growth data, trait data, and are those data available? In the medical meta-analysis field, this corresponds to the PICO criteria. So sometimes I see people call PICO criteria in ecology meta-analyses. It's the same thing. Populations, interventions, comparators, outcomes. Those are the things I just listed. This is how we define the types of studies that we want to include in our review, right? Okay. If you don't feel confident setting the scope, that probably just means you don't know the literature well enough yet. So read more, work with people with more experience, and they can usually help define like what's reasonable, like how big are we talking about or how narrow. Okay. And then as you're deciding on this scope, you're building your inclusion criteria. And most reviews and meta-analyses report this as a list. Studies were included when they met these criteria, 1, 2, 3, 4, 5. It really is that formal. So you want to make this list early about exactly what it is you're using to decide that a study is suitable or not suitable. And this also helps you think about the metadata that you're going to be tracking when you actually extract the data. The metadata are the factors that you think are likely to explain heterogeneity across studies. That's the contribution of the meta-analyst. We're adding the metadata. We're coding that. It's what we think could be why some studies find this and other studies find that. So we're adding that when we extract the data to build our metadata set that includes the raw data, but now all the metadata that we think is likely to matter that is defined by our inclusion criteria. Okay. Once we have all that set, we're ready to start looking for papers, right? So defining our search terms is like any experimental study, going to benefit from some piloting. Right? Get into Web of Science and try some stuff out and see what gets you the kind of studies you're thinking about. And this is where people do a lot of like adjusting of their scope. They may think that they have this particular set of terms that reflects what they want to look for, and it returns 30,000 papers. And they're like, what? I can't do that. So then they adjust the scope and the search terms, and they're trying to find something feasible. This is how you end up with 30 papers. You're adjusting, but this is all piloting, right? As long as we're taking notes on it, this is how it works. Obviously, we use Boolean searches because we're trying to connect multiple search terms. I'm showing you an example down here at the bottom from a meta-analysis in my lab. And these are the kinds of notes that we take. So we have the date, where we did the search, the years that the search includes, the search query itself, and how many papers it returned to us. Those data actually need to be reported in the paper. So you want to record that. If I do a bunch of these searches, I make all these notes, and then I circle the one that I'm going with. And I document all of this because I actually have had reviewers ask me how I landed on search terms. And I was like, I don't know. And then I went back to my notes and I had everything. So this is good to have good records of. Where are you going to do your searches is also important. And there's like each of these things I'm pointing out, there are papers about. We're not just making this up. Somebody has investigated this. And in the case of search systems, there's this super useful paper with these extensive tables of criteria for why some search engines are better than others. They searched 30 different search databases, and then they assess them on all kinds of things, but mostly precision, recall, and reproducibility. Reproducibility, that's an important word in science, right? We want to be transparent enough in our methods that it could potentially be reproducible by somebody else. Same thing for systematic review methods. And what they found is that Google Scholar is a stinker. Don't use it as your primary search engine. It fails all of these important criteria. They say, okay, you can use it as a secondary search database, but don't make it your primary because it fails too many of these tests. Web of Science is their main principal search system that is good in all of these regards. I think that's really important because it's so easy to use Google Scholar that we tend to use it, but it's not very good. And their other recommendation is that you should always use 2+ databases in systematic review. Oh, what a pain. I hate that recommendation myself. I like Web of Science and then I would like to be done, but no. You have to decide if you're going to include only peer-reviewed publications or also gray literature. You have to decide this up front. Are theses, government reports suitable for you? What about unpublished data? A lot of people want to use unpublished data because they're probably sitting on a lot of unpublished data, or they have friends working on the same things that have unpublished data. You shouldn't do that. That's how you introduce bias to your data set. Unless you're going to do a global call to the community, if anybody has unpublished data that want to share it with me for my meta-analysis, that's great. If I just use my own or my friends, now I've made it subjective, right? That's not good. So I, as a general rule, do not use unpublished data in meta-analysis. Some people do. You just have to be transparent about this. And then, of course, here's where language comes in. And there's a really nice paper that actually just came out like 2 years ago about language and systematic reviews. And they did all of this assessment into why there are language barriers. I mean, beyond the obvious. But they found 2 suggestions that they thought could help fix this. One, of course, is funding, like everything. If we had more funding to pay for translation services and more time, people would be more inclined to go to the effort to include non-English speaking papers. And the other is bigger teams with a diversity of languages represented, also linked to needing more money. So basically more money. But I appreciate that they're addressing it because this is a major problem. And I think it is so cosmopolitan now in science, and we have Zoom and all these ways of interacting with people around the world. We should be putting together international teams with expertise in different languages. That would really help. Okay. Now you've done all this, you've run your searches, you've got your pile of papers that you want to assess. The first assessment is usually just of the abstract. So you make a first pass through the abstracts to decide yes or no. This should be pretty fast, right? I mean, I think it is. To me, it is. I just make it fast. We just cruise through it. We have our inclusion criteria. We look at the abstract. If there's anything that's a clear miss, then we put it as a reject and we put why. Which inclusion criteria it did not meet. If I cannot tell, or it's a yes, I just code it as a yes, or I can't tell for further assessment. This is a way of fast rejection of the ones that are clearly not good fits. Usually you can tell from the title. Okay. Once you've done that, you have your list of papers for full assessment. And the full assessment, well, here's how we do it in my lab. We do the first assessment with, we just export everything from Web of Science into an Excel spreadsheet, which includes the abstract. We read the abstract, we code it yes or no. If we give it a reject, we tell why. Which inclusion criteria did it not meet? Then we have all the yeses or the maybes, and we download those. And now we have to read the whole paper, and we determine whether it fits our inclusion criteria that usually have to do with the data itself. So, and the methods, like the methods are really critical to read here to rule out things like pseudo-replication, using instruments that are now considered to be sort of like poor ways of measuring something, etc. This takes longer, obviously, but this is what yields that like final set of studies that you are confident have the data you need and are basically like you're going to be your data points. Sometimes I see people just squandering or, stuck in this step forever. I think it's best to like, if you have your inclusion criteria as a list or a template that you can copy into that sheet, so you just mark it what it's not meeting to try to make this efficient, that really helps. This shouldn't be the slowest part. This is like what we do. We put those inclusion criteria into metadata. So we have this in the spreadsheet itself as different columns. I'm just showing it to you transposed so it would be easier to see. But we've set it all up so we know what we're looking for. And that helps us rule things out and also helps us sort of organize the paper right away into these different kinds of metadata categories. Okay, these are just examples. And then when we have filled it in for a paper, we're coding the metadata for each of these different categories, and we're also identifying where the suitable data are in the bottom. So we like write down actually where the data are that we're going to extract. This is our assessment of the publication. This is probably a pretty slow step, but it's crucial to do in this kind of organized way to be sure that we're being consistent across papers. I would also say this is really important if you've got multiple people doing assessment and extraction, because you want to all be doing the same way. Just like if multiple people were collecting data in the field, you don't want error because of who was collecting the data. So the same thing for meta-analysis. Okay, these are just examples. Now, let's say you do this and halfway through, you're like, oh, now I see they're calling this thing that I thought was called this something else, and I might be missing a whole group of studies, or I'd like to include this other kind of thing. So the question is, can you change your inclusion criteria? Oh, wait. Yeah. Can you change your inclusion criteria midstream? This is called mission creep. Like, it's got a formal name in systematic reviews. It's generally frowned upon because if we change our inclusion criteria midstream, we've now got two separate sets of inclusion criteria, the sort of before and after. And so, I mean, I feel like it's almost inevitable something changes midstream, but I have to then go back and redo everything quickly just to make sure I didn't miss anything, so that they all are going to meet ultimately my last set of inclusion criteria. So it's a pain. This is why it's actually worth it to take a lot of time in the beginning and really pilot and make sure you're confident about your body of evidence and your inclusion criteria to limit how much of this you have to do later. And I know lots of people are like, oh, don't worry about it, let's just keep going. But again, there's like whole papers on why mission creep is a problem. Ultimately, we're going to compile all of these records of assessment so that we can report things like the total number of returns from your searches, how many of those passed, how many were rejected, and why. And that gives us a measure of sort of how good your search terms were, right? If you ended up with few rejections and a lot of passes, it sort of indicates that probably you could have broadened those search terms. You may be missing things. On the other hand, if you have a search where you had to reject 90% of the papers, it was probably too broad a search and you probably should have narrowed it. These are good indicators of sort of how reliable your search methods were. Okay. You can, of course, do additional searches, but you just, like everything, want to record this again and go through the same process. I always do a final search. Like if I do a search, and then we were doing assessing it, and we're extracting all the data. And by the time I'm ready to do the meta-analysis, it's been like 2 years. So I always do a final search there to catch anything that came out in those 2 years I was working on it, to just catch it up. And I would just record that. It's like a catch up. Oh yeah, there it is, catch up searches. The other really useful and typical thing to do are called reverse and forward searches. So for all my papers that are positives, that pass all my inclusion criteria and that have the data I want, I will check their references. And their references are usually really good sources of more papers. Those are reverse searches. And I track those down. Tyler's helping me. And we do that until we've reached sort of saturation. Forward searches are for any really good papers, especially old papers. I look at everything that has cited it. And that catches some things often that I didn't detect with my main searches. Those are the forward searches. That's also really useful and productive. Also useful for review papers. Like if I find a review on my topic, I do a forward search. I see everything that cited it. That's usually good. All of this needs to just be reported. For meta-analysis, the ultimate goal is to have everything on your topic that's suitable. These days, because the literature is so big, I think people are like, well, that's impossible. But as long as we have really good inclusion criteria reported and transparency in your methods, we know what you did. That way we can assess, does it seem like they caught everything, or is it likely they missed something, and here's why I think so. That's the whole point, transparency. Okay. Here's all of those things. Reverse and forward, final catch-up searches. Now you just sort of like synthesize those data. Phew. See what I mean? A lot of a meta-analysis is just getting a good body of evidence. It really is most of the work. Okay. And these days, to facilitate this, there are actually pretty good formal reporting programs that help us organize all of this information. So you may have heard of Prisma. That's a really popular one. You can run Prisma on your meta-analysis, and it will just make this really nice schematic showing how all your search terms and how many returns you got and how many of those are positives and how many you got in reverse and forward searches, etc. It just organizes all of it for you, but you need the pieces to feed into it. Some journals require Prisma. Some journals require ROSES, which is another sort of format, standards for systematic evidence syntheses. This is why I recommend you look at the target journal early so you know exactly what kind of information they will require upon publication. Okay. So now you have your body of literature. Are there any questions at this point? Because I feel like that's a lot. Yes. You know you ran a good pilot because when you do your reverse searches, you're not finding much, right? What you want is a search term or search query that's catching everything you want it to catch. If you're reading the papers that you catch and it's like tons of them in the references that you didn't catch, you're like, how did I miss all these? What words are they using that my search term didn't detect? That's why I really think reading some papers early is helpful, because you may not know that different ways that people refer to the same thing. But I don't worry too much, because the reverse searches are easy to do and they're super productive. All we're doing is depending on all those other scientists to have found good studies relevant to their own study, right? They're helping us out, and that's good as long as we reach saturation. So at some point, you should be looking at the references and there's no new ones. You've caught them all. Then you know you've got it. And some people are more thorough than others. And I think it's getting slippery. Like when I was first learning this, it was like, you have to get everything. Keep working. And now it's like, there's too many papers. We can't include everything. So I think that's inevitable, because it's true. There are too many papers. But that's all the more reason why we need to be transparent about how we found the papers we're including. So these methods of systematic review and reporting it is just so helpful to the reader, so they know about any gaps in what you're synthesizing. Good. Any other questions? All right, so now what do you do with this big pile of data? Well, the first thing you should do is decide if you're doing a meta-analysis or not. Because if you're not doing a meta-analysis, it's way easier. You just read those papers and you write your review, right? And that can be really useful. Like I said, synthesizing across fields. Maybe you're trying to elucidate the value of a new approach. Maybe you're trying to argue for something. I don't know. Anyways, I think I've done a lot of these kinds of systematic reviews, and I really like them. Meta-analysis is especially useful, like I said, when there's no consensus. And it's also really useful when the body of data are sort of consistent enough in how they were collected or are presented that I feel comfortable analyzing them together in a big data set. If it's too variable how people collect data or test certain things, then I'm not sure that analyzing them together is really valid. I'm putting apples and oranges into the same model, which I don't know. People have different ideas about that. I think if you think there's consensus in a topic already, it really isn't worth the meta-analysis, because it's not interesting. There's nothing new elucidated from that. You've just done a lot of work to confirm what everybody already knew. That maybe you want to do that. I don't know why. It feels like a lot of work. But meta-analysis is super useful when it's unclear why some studies find this and other studies find that. And you want to know, or you have ideas about, well, it's because this was in the tropics and this was in the temperate, or it's because they were measuring these traits and they were measuring those traits. Then meta-analysis is helpful because it can analyze those sources of heterogeneity across studies. And then it's worth the cons, which are it's hugely labor intensive. And if you don't have a good solid body of evidence, there's this false sense of objectivity or generality. I think that is one of the biggest risks, but it's also one of the biggest temptations. We do meta-analysis because it is high impact and it carries this sort of like sense of objectivity and generality, but whether or not that's valid is sort of why we need to assess it by reporting the evidence. And again, this book is so helpful. It really goes through all of these topics. It's a great resource. And then I also listed down here some other folks in the community who are I think, real leaders in this, especially in ecology and evolution. And Marc Lejeunesse in particular has this really great YouTube course where it's many different it's like a series where he goes over so many issues, statistical issues, systematic review issues, and he's just so clear. I think that's a really great resource. And then Weichbauer, probably not saying that right, he has written the R package that most people use, Metaphor, to do meta-analysis. And he also has really good blogs and tutorials. So those two people stand out, but there's lots of great resources and folks working on this who are the real innovators, who are really helping us do this better, I think. Okay, so let's say you decide you want to do a meta-analysis. Here's how you would do that. First, you would decide what's the suitable effect size. So what's the nature of the effect that you mean to be synthesizing? Is it group differences? Is it a correlation? That just is going to determine then which data you extract from the paper. Then you actually extract all the data. Now you have to homogenize this compiled data from all these sources, which includes things like making sure they're using the same units for things. And that can be a painful step. Then you're sort of setting yourself up for the model. So you identify your predictor variables. Those are the ones that you think may explain heterogeneity across the studies. You have to code for your random variables, which includes things like phylogeny. So phylogenetic meta-analyses are the standard now. We take into account the non-randomness among species. And also the data sources should be coded as a random factor. So the publication itself, if you have multiple effect sizes come from the same publication, they're not independent of each other. So you want to account for that by including publication as a random variable. Then you run your models, you get your group means, you get your confidence intervals, and now we get our answer. So what we're doing with meta-analysis, remember, is we're trying to synthesize an effect of something, like a treatment, to use our medicine example, our treatment, or in the case of my work, like drought effect on plant survival, something like that. So we're synthesizing this effect across many studies. So the effects are the replicates, and each publication that is suitable for your meta-analysis probably has multiple effect sizes or effects that you can extract, right? Because they tested multiple species or multiple traits or multiple sites or multiple metrics of performance or traits or whatever. So each of those is its own independent effect. It's a data point in our metadata sets. So we're compiling this big database. Each row is one effect, one response to a medicine or one response to a drought treatment or whatever. So what kind of effect size you are going to be using will determine what data you pull from the paper. So that's the point, really. I guess I'm coming to it. One of the biggest issues is data availability. So when I can just mention this really quickly. For group comparisons like treatment versus control, we're pulling the means, the standard deviations, and the sample sizes of our groups. And that goes in my one row because they're going to be used to calculate the one effect size, the difference between the groups, weighted by the variance in the sample size. For a correlation kind of effect size, I'm pulling the correlation coefficient and the sample size. And that are the data that I have to extract. So figuring out your effect size is important so you know what kind of data you're looking for in the paper, right? Okay. Now, where are those data anyways? So the easiest thing is if they've archived the raw data or even group means or whatever, and we can just get that ourselves from wherever they archived it. Of course, that's really common now. Lots of people are sneaky and they archive their data as like, I don't know, least square means or community weighted means in formats that we can't use for meta-analysis because we actually need the raw data. So I think people know that, and that's why they do it. So they're like archiving their data, but not in a usable format. Old publications, it's never archived, right? So we can't, that's only sometimes useful. But when there is raw data, or when we can extract it from the paper in, say, like a table or a figure, we can do that without restriction because data are not copyrightable. So data are viewed as facts, and facts cannot be copyrighted. This is why we can extract data from publications and then reanalyze them ourselves, because the data are not protected by copyright. The model output, the analyses, the graphs, the writing, all of that is copyrightable. I cannot use any of that without explicit permission, but I can just scoop the data because of this loophole. There are some cases in Europe where people have argued that the data are copyrightable because they were behavioral data that they defined and coded and like took through cameras, birds in particular. But most biological data, genetic data, trait data, performance data, structure data, that's not copyrightable. Interestingly, when I pull that data and I create a metadatabase with new metadata that I've added intellectual property material to that. The database I create, the data are copyrightable. What a weird thing, right? So when you publish a metadata set, the database can be copyrighted. People can't reuse it without it being open access. So we have to be really careful about that. But otherwise, we can just pull the data from a data paper. Whether or not that's ethical, there's a lot of discussion about this. And some people are pushing for getting permission to use data just because it's ethically better. And I think that there's some real validity to that. And this is something that's sort of contentiously argued right now in this field. So the problem is people want to use the data for meta-analyses without having to ask every author, because that's really hard. But just because things are hard doesn't mean we shouldn't do them. So I would say this is a part of this that's sort of uncomfortable and that people talk a lot about, and that you should decide what feels best for you. I will finish this point just by saying, you know, there are, I would argue, separate data sovereignty issues associated with indigenous and cultural knowledge data. So that is clearly established. Anything that is cultural data is clearly covered with data sovereignty, and we should seek explicit permission to use that. What's less obvious, because it hasn't been clarified, is whether, like, for example, biological data collected in an indigenous place is also covered in that same sort of way. I think people would generally view that as part of indigenous data sovereignty, all biological data, but it's currently sort of excluded from this expectation of getting explicit permission to reuse. In general, the reusing of data is not as far along in indigenous data sovereignty discussions as collecting new data are. We have pretty good ideas of how to collect new data in ethical ways. Reusing old data that was not collected in ethical ways is murky. But I think people are really talking a lot about it, and hopefully this is going to get sorted out soon. Of course, we're all tempted to use automation tools to extract data and make our lives easier. And if your particular kind of data and your set of papers are conducive to that, then you should. That's great. This could make it a lot more efficient. I'm not familiar with those kinds of scenarios because in ecology, it's so variable across studies how people collect and present data that it's not easy to automate it. And we've tried. So We actually just do it all manually, and I'd say that's the standard in ecology. But maybe now that we know that these automation tools are sort of in development, that people will move towards more systematic data collection and presentation. I'm not sure, but we're not there yet. Maybe molecular studies are further along, and collecting their data in a way that is conducive to this. For us to extract the data, we actually mostly pull it from graphs. So it does end up looking kind of like this. It's either in a table or it's in a graph. And if we're doing a group mean comparison, like I said, we need the mean, standard deviation, sample sizes. Ultimately, we're going to use those to calculate effect sizes. And the two most common are Hedges D and log response ratio, each of which has a corresponding variance component. So that's the measure of accuracy and magnitude that we're after. For correlations, it's Pearson's R and sample sizes. So those are the sort of most common types of effect sizes in my field. But that book by Karicheva has a whole chapter on effect sizes. So depending on your question, there's a lot of different effect sizes you may be interested in. Like I said already, if it's in a table, if they report the means and standard deviations in the table, that's so easy. So that's great. If they've archived it, that's easy, and that's also great. If it's in a graph, that's a big pain for us, but we can do it. Lots of times, especially old papers, the data aren't reported, like they just report the statistical output. And then this is a sort of point of decision making for you about whether you want to try to track down the unpublished data or the data that were published but not in a way that you can use to the authors. My very first meta-analysis when I was a postdoc, I did contact the authors of papers where the data were not presented in a way I could use. And I probably contacted 50 authors and I got like 2 or 3 replies. So I don't do that anymore because I don't think it's worth it. And I just write as an inclusion criteria, the data are presented in a usable way. Then I've sidestepped it. But it depends on your question and how much this could be a problem for you. If it's a big proportion of your studies, it may be worth tracking down, if you even can. To extract the data graphically, we use WebPlot Digitizer, and Yulia Kricheva did a survey maybe 5 years ago of folks in the ecology field who do a lot of meta-analysis, and this was the winner. And I agree, it's really easy to use. And it's free. It's just a web-based tool. And you basically just take an image of the figure and upload that, and then you can calibrate because of the axes labels. And then you can estimate the mean and the error bars, and you download that as a CSV. So it's really tedious, but it's not difficult. And then ultimately what you get is a metadata set where each row is going to be one effect size, right? It's one species with their one group mean comparison, includes all the metadata factors that I think might matter, like growth form and biome and habitat type in my case. And now, once I have this big metadata set, I'm ready to actually run the meta-analysis. So of course, this is like 95% of the effort is to get to this point. Once we get to this point, we're ready to cruise. So how do we actually run the models and analyze the data? Well, I use Metaphor, which is an R package by that Weichbauer that I told you. This is what I see most people using, although there are other packages out there. And you can actually, you don't even need to use a Metaphor package at all. And sometimes I just use Metaphor to calculate the effect sizes, and then I do mixed models like I would with any kind of data. I just do a weighted by the variance mixed model. So there's lots of flexibility here. And I think if you're new to this, the thing to do is to find some recent meta-analyses that are sort of similar to what you're doing and look at their code and see how they did it, especially by those people that I mentioned before who are really good at this. But essentially what you do is you calculate the effect size based on those raw data, which will give you, like, for a group comparison, log response ratio or Hedges D and the associated with in-study variance. Then you can run your random effects model, which is sort of the pooled overall effect. That's where there's already consensus, all you're doing is confirming that pattern that you already knew. But if you think there's a lot of heterogeneity in your data set, then the random effects model isn't that interesting because it doesn't include any fixed factors yet. The whole value of this is to add in those fixed predictor variables that could then explain heterogeneity across the studies. And those are formally called heterogeneity tests. So those are mixed models because it includes the random factors of publication and phylogeny and maybe other things that you think are random, but then also includes the fixed factors, the drivers of heterogeneity that you're after. And from those, you can ask it to produce or report, the group estimates with 95% confidence intervals. And then you can sort of graph that like this, and we can see those group differences. Say in this meta-analysis, I was looking at how traits varied as leaves develop. So when they're really young leaves to when they're fully expanded and mature. And a negative log response ratio means that they declined during maturation. So nutrient content and secondary chemicals decrease in concentration from young to mature leaves. But physical traits like leaf toughness or prickles and indirect defenses like extrafluoronectaries increase as the leaf matures. So I could detect statistically significant variation among those classes of compounds using a heterogeneity test. And then those are those group estimates with the 95% confidence intervals. And the way we interpret 95% confidence intervals is if those do not overlap with 0, that's a statistically significant difference between, in this case, actually, it was young leaves versus mature leaves, right? That was my group comparison. So there's my heterogeneity test. QM equals 4.4 degrees of freedom, 428.05. P-value is really significant. Okay. Oh, and see, the code is like super simple. That's what I mean. Once you got this ready, it takes like a few minutes. Then you've got your answer. Now you want to interpret it. And I'm just putting this in as a word of caution because I just see too often that people extrapolate way beyond their inclusion criteria. Your inclusion criteria set the scope. So we should be interpreting within that same scope and not just like across all plants everywhere. In my case, all plants everywhere. So you need to be aware of that scope. And I think when interpreting multiple sources of heterogeneity, they may not be independent of each other. They may be confounded. So looking like explicitly at non-independence among your predictor variables is really important. For example, what if like I have it here. What if the herbs and forbs were all sampled from temperate regions, but woody species were all sampled from tropical regions? My growth form heterogeneity test is significant. My biome test is significant, but you see how they're kind of redundant? So I want to know that. So I look specifically at non-independence among my predictor variables, just so that I can interpret this in a sensible way. And then the final thing is that there's usually expectations of formal tests for publication bias. So the easiest way to do this is with a funnel plot analysis. This is what a funnel plot analysis looks like. So theoretically, it should look like that funnel on the left. So the y-axis is standard error, which is a kind of proxy for sample size. And then the x-axis is the effect size. Like in my case, it was log response ratio. So the way it works, and we can look at, this is from my actual data. This is the mean log response ratio. And this is a measure of sample size. So the studies with big sample sizes should have effect sizes close to the mean because they carry more weight. That's the whole point of having a big sample size. And you can see all those big studies do cluster near the mean. The small studies should be scattered on both sides of this sort of randomly on both sides, a positive and negative direction of the mean. And that's what you see here. Studies that are smaller in their sample size, so they're less accurate, are on both sides. That that symmetry indicates that there's no bias in the directionality of the effect to get published. If you have asymmetry, do you see how there's more studies on this side that are small than over here? That indicates publication bias for a positive effect. Because when they're smaller, sort of less quality, they're still getting published if they confirm the hypothesis. But a negative result, they're less likely to get published. And I can do a statistical test of asymmetry, which I did, and the p-value is 0.0145. And that gives me some confirmation that this meta-analysis is likely affected by publication bias. There's nothing I can do about that. But being aware of it is really helpful. Because if I want to conclude that there's some generally positive effect, I also want to condition that by saying, but there also appears to be a publication bias for studies that confirm that. If you don't do a funnel test, you're probably going to get the paper bounced. I've seen that several times. You have to formally assess publication bias, at most journals at least. Okay. One of my favorite reasons for doing a meta-analysis is when I suspect there are data gaps. Sometimes I go to the trouble to do a meta-analysis I know the answer for just because I want quantitative evidence that there are gaps. And this is really helpful. So you may elucidate that the vast majority of studies are in the tropics or the temperate or on woody plants. There's a major bias in what we're choosing to test in this particular question, whatever it is. I want to be able to recognize and identify that data gap because it limits how confident we are to generalize about whatever our answer is. But most importantly, it provides future research directions. And to me, that's one of the best things of meta-analysis is to highlight something that seems like maybe an important source of heterogeneity, but we're not sure and there needs to be more studies. That's the best thing. Finding more things to study as a consequence of meta-analysis. So we really spend a lot of time inspecting our metadata set and looking at different kinds of replication across our different sources of heterogeneity factors to identify where we have a lot of weight of evidence and where we're really lacking in evidence. That can be understudied taxa. Sometimes there's an overemphasis on model systems, which is really annoying to me. There are certain traits that are really neglected. In our case, it's like root traits are really neglected. Sometimes there's forms of data, indigenous or biocultural data, that may be suitable that's not incorporated because of some kind of bias. There's usually biogeographic bias that's really big. And this happens a lot, and people are starting to crack down on it because there's a lot of papers where the title will say things like, there's a global trend in something. And then they've done a meta-analysis, and they have studies from every continent, say, but when you look at the weight of evidence, there's like one dot in Africa, right? So no, that's not a global pattern. That's a biogeographically biased pattern. And we need to acknowledge that so that we can find ways to support publications from those underrepresented places, right? So I think that's a really good use of meta-analysis. And not because we're trying to promote parachute science, but because we're trying to figure out ways to lift up those local scientists in places that are underrepresented. And also, are there tools and approaches that aren't available to people all over the world? So that's another thing that comes up a lot, is sometimes you're doing a meta-analysis on a topic that requires some really expensive equipment that's hard to get, and that explains a biogeographic bias. So these are all important things to consider, and I think meta-analysis can really help us argue for them. Okay, I think I'm like almost done, which is great because I know this is a lot, but I just wanted to end on one like tempting little tidbit, which I think is really underutilized. Cumulative meta-analysis is just a meta-analysis where publication year is a variable. So what we're doing is we're looking at the pattern of evidence over time in a body of evidence. And Yulia wrote a paper on this like over 20 years ago, Cumulative Meta-Analysis, a new tool for detection of temporal trends and publication bias in ecology. It was a new tool 20 years ago, and like no one ever uses it. It's not anything different except that you're adding in year, but I really like it because it can show us how often in early days of a topic, the effect sizes are positive and minus. Like they're bouncing around a lot in the meta-analysis because people don't know yet what's going on. And then people start to sort of like have an idea of what's going on, or maybe there's just a weight of evidence and it either kind of goes a positive effect or a negative effect. And it's sort of stuck there for a while. And sometimes it's stuck there forever. And sometimes there's a paradigm shift and it becomes something else. And that's hard to see except in this temporal cumulative meta-analysis way. And then she argues in this paper that it's also really good for detecting publication bias. So sometimes you see it start to stabilize in a positive because of a lack of publications contrary to that hypothesis. And then again, sometimes over time, people start to argue against that because they think maybe they're neglecting negative evidence. And then you can see a paradigm shift as a consequence of removing a publication bias. I like it, but I almost never see it being used. So I just wanted to throw that out there because to me, it seems like kind of a exciting and underutilized tool. And then I think I just have my take-home messages, which is I hope this is inspiring you to do a meta-analysis. It is a lot of work, but it's really satisfying. And I do think you get high impact papers at the end of the day. My most well-cited papers are my meta-analyses. In general, though, even if you're not going to do a formal meta-analysis, you should still do a review. It's a great way to become an expert on a topic. It is a good way to like get new insights about something. That's one of the reasons why I think it's really important to read all the papers and not just extract the data and move on. Because a lot of the times what we're trying to do is find new ways of thinking of things and new interpretations of the same patterns. And that only comes about from a lot of reading, I think. So this is a good way to just do a ton of reading, but have it yield something at the end of the day. And ultimately, if you have these skills, I think they're useful to other people too. I've had lots of people ask me about meta-analysis. And so it's a great way to get new colleagues and new collaborations, and that's always fun. So good luck. And please do this survey that Shivani sent me before you go to reflect on this. And there's my email address. I usually have a big bibliography of papers that I have found useful on meta-analysis and ecology and evolution linked to my presentation, but I noticed before this that it's not linked in there anymore. If you want that, email me and I'll send it to you. And I'm also happy to answer questions. Thank you. Yes. Sure.
I was wondering if we can use a systematic review to also review knowledge gaps and evidence gaps.
Yes.
Okay.
Systematic review is just the formal set of methods for how you identify the body of literature you want to review. And so anytime you write a review, I would say anytime you write a review, you should use systematic review methods. Usually those go in supplementary materials. Like I just reported, I use systematic review methods, supplementary materials. In a meta-analysis, it's in a method section. But just to like highlight knowledge, like in a more of a short review, like a mini review.
Of the review is to highlight what we don't know and what we have tons of evidence. Like I work with coral reef, so we have tons of evidence, field work, and also mathematical models. But I don't know if they are supporting the same stuff. So maybe I have like this specific approach, just supporting this pattern, and the other they don't like.
Well, that seems to me like especially important that you use systematic review methods because you're trying to convince people that those studies don't exist. But how do they know you've searched exhaustively? You need to tell them how you've searched. That's the transparency. Like, this is what I went through. These are all the steps I went through to identify studies. And here's what I found. there are clear gaps here. Yeah.
Question in the chat from Veronica. Since making theses of well, how do you come up with proposal or funding ideas for meta-analysis? And as a follow-up question, is it different than for experimental proposals?
You know, it is different. There aren't a lot of good ways to get funding for synthetic research. NSF has a little bit of funding. They have one program, I can't remember what it's called, for synthesis. It's a small amount of money, and you have to sort of be like mid-career. So it's not useful for people in postdoc. NC's in California is a synthesis center, and that's all. They've got lots of postdoc funding, and that is a good way to get synthesis funding. My postdoc was just a regular NC's fellowship, which is like the UK version of NSF, and that so it's just a normal postdoc fellowship. And then there's a center in Montpellier, France called CESAB, and I've gotten a couple grants from them, which allowed me to spend some summers in Montpellier doing synthesis. They are an ecological synthesis center. So that's the thing to do, is find a synthesis center and apply for funding directly to them. In the US, NC's is the only one I know about, but in Europe, there's a bunch of them. Like I said, they're doing a lot of this kind of work in Europe. Yeah. But I think in general, there's less funding for synthesis than research, than like active data collection based research. I don't know why exactly.
If there are any other questions from folks on Zoom, please feel free to unmute yourself, or you can post a question on the chat.
All right. Well, thanks everybody.